Hello everyone, I hope that you're all doing well. My name is Richard and welcome to Group Advanced Concepts presentation number 5. So just a quick description. So cellular respiration is the process that allows your body to harvest a huge amount of energy from a single glucose. In fact, it's so efficient that you get 30 ATP for every 1 glucose molecule you eat. This exercise is going to break down exactly where each ATP comes from. So our goal for this week, we are to walk the audience through every step that is involved in generating an ATP, NADH, or FADH2, including the names, structures, and enzyme for each step. We're also going to look up the conversion rates from NADH and FADH2 to ATP, and calculate exactly how 30 ATP are formed. And finally, we're also going to include any steps which uses an ATP, as this will count against the total ATP formed. So this is an overview of cellular respiration. As you can see based on the image over here, it consists of 4 important steps: glycolysis, pyruvate oxidation, citric acid cycle or Krebs cycle, and oxidative phosphorylation, which includes the electron transport chain and ATP synthase. So let's start with step 1, which is glycolysis. So glycolysis takes place in the cytoplasm and it occurs in 10 steps. So I've broken it down so that these are steps 1 through 5. So you can see first that in step 1, the phosphate group is transferred from ATP to glucose, making G6P. ATP is consumed, as you can see. In step 2, G6P is converted to its isomer F6P, fructose 6-phosphate. In step 3, phosphate group is transferred from ATP to fructose 6-phosphate, making fructose 1,6-bisphosphate. And ATP is again consumed similar to step 1. In step 4, F16BP splits to form 2 3-carbon sugars, as you can see here, dihydroxyacetone phosphate, DHAP, and glyceraldehyde 3-phosphate, G3P. So from here, DHAP is going to be converted to G3P, and even though both of these molecules are isomers, it's the glyceraldehyde 3-phosphate that can continue through the next step of glycolysis. Moving on, step 6 through 10. So two half reactions are going to occur simultaneously. So glyceraldehyde 3-phosphate is oxidized, and NAD plus is reduced to NADH plus H plus. This is an exergonic reaction forming 1,3-bisphosphoglycerate, as you can see down here. In step 7, the 1,3-bisphosphoglycerate donates 1 phosphate group to ADP to form ATP plus 3-phosphoglycerate. In step 8, the 3-phosphoglycerate is converted to its isomer 2-phosphoglycerate. In step 9, the 2-phosphoglycerate loses a water molecule and it forms phosphoenol pyruvate or PEP. And finally in step 10, PEP donates a phosphate group to ADP and it forms ATP and pyruvate. So note that reactions 6 through 10 happen 2 times, because the previous step, step 5, left us with 2 molecules of G3P. So we had 4 ATP produced from steps 7 and 10, because it happened twice. We're subtracting 2 ATP consumed in steps 1 and 3, and that leaves us with 2 ATP overall at the end of glycolysis. And we also have 2 NADHs that are formed from step 6, as you can see here. So the net products for glycolysis include 2 pyruvate, 2 ATP, and 2 NADH. So from here, we can move on to pyruvate oxidation. This up here is the general pathway that occurs. So you have pyruvate. It's going to go under decarboxylation, oxidation, transfer to CoA to form acetyl-CoA. So if you recall, in last week's presentation, we went into more depth for pyruvate oxidation. So I'm just going to give a shorter version of what's going on here. So it takes place in the mitochondrial matrix. And you can see here, in step 1, a carboxyl is removed from pyruvate, releasing CO2. A 2-carbon molecule is left behind. Step 2, this 2-carbon molecule is oxidized, and the electrons lost in the oxidation are picked up by NAD+ to form NADH. And the oxidized 2-carbon molecule in green in step 3, it's attached to coenzyme A to form acetyl-CoA. And the job of acetyl-CoA is to carry the acetyl group to the Krebs cycle. So the net products for pyruvate oxidation include, again, 2 acetyl-CoA, 2 NADH, and 2 CO2. Moving on from pyruvate oxidation, going to step 3, which is the Krebs cycle. So there are 8 steps in total, and this looks a little daunting, but let's take it step by step. So in the first step, acetyl-CoA plus oxaloacetate forms the citrate, which is over here, using the citrate synthase. So it's 2 steps technically. In step 2, the citrate is converted to its isomer, isocitrate. This is 2 steps, not step 1. There's a removal and then an addition of H2O. In step 3, the isocitrate is oxidized and releases CO2, leaving behind alpha-ketoglutarate, as you can see here. NAD+ is also reduced to NADH in this step, as you can see down here. In step 4, the alpha-ketoglutarate is oxidized, releases CO2, and reduces NAD+ to NADH. Again, very similar to step 3. So the remaining 4 carbon molecules picks up CoA, forming succinyl-CoA, as you can see here. In step 5, the CoA of the succinyl-CoA is replaced by a phosphate group. It's then transferred to ADP to make ATP. And in some cells, GDP is used instead of ADP to form GTP, and succinate is formed in this regard. So just on an aside, GTP is similar to ATP. They both serve as energy sources, and the two can be readily interconverted. Which of the 2 molecules is produced during the citric acid cycle depends on the organism and cell type. So for example, ATP is made in human heart cells, but GTP is made in liver cells. So going back here, in step 6, succinate is oxidized, and that forms fumarate. And you can see here that 2 hydrogen atoms are transferred to FAD to form FADH2. And the reason why FAD is used here is because it's a better electron acceptor than NAD+. In step 7, water is added to fumarate to convert it to malate. And finally, in step 8, oxaloacetate is regenerated by the oxidation of malate, as you can see here. And again, NAD+ is reduced to NADH. So, in summary for the Krebs cycle, in one turn of the cycle, 2 carbons enter from acetyl-CoA and 2 molecules of carbon dioxide are released. 3 molecules of NADH and 1 molecule of FADH2 are created, and 1 molecule of ATP or GTP are produced. And since we have 2 molecules of acetyl-CoA from pyruvate oxidation, we multiply the top numbers by 2 to get our total net, 6 NADH, 2 FADH2, and 2 ATP. And finally, step 4, which is oxidative phosphorylation. Again, this consists of the electron transport chain plus ATP synthase. And this takes place in the inner membrane of the mitochondria. So you can see here that the NADH and FADH2 from previous steps transfer their electrons to the molecules near the beginning of the transport chain. And they also turn back into NAD+ and FAD, which can be reused in the previous steps. The electrons move from high energy to low energy, and it releases energy. And some of this energy is used to pump H+ ions from the matrix, down here, into the intermembrane space, up here. An electrochemical gradient is established at this point. At the end of the electron transport chain, the electrons are transferred to O2, which splits in half and takes up H+ to form water. And as H+ ions flow down their gradient back into the matrix, over here, it passes through something called the ATP synthase. This harnesses the flow of the protons to synthesize ATP. So again, there are 2 important functions of the electron transport chain. 1 is to regenerate electron carriers, and 2 is to make a proton gradient. So why are these important? In terms of regenerating electron carriers, you can see here again, NADH and FADH2 pass their electrons to the electron transport chain, and they turn back into NAD+ and FAD. And if you recall from earlier slides, these NAD+ and FAD are used in glycolysis and the Krebs cycle, and must be available to keep them running. And in terms of making a proton gradient, you'll notice there's a higher concentration of H+ ions in the intermembrane space, and there's a lower concentration of H+ in the matrix. This gradient, also known as a proton motive force, represents a stored form of energy that can be used to make ATP. And then going on to forming that ATP is known as chemiosmosis, also defined as the energy from a proton gradient used to make ATP. So ATP synthase is turned by the flow of the H plus ions, as you can see as it goes down here. So as the ATP synthase moves down the electron chemical gradient, it turns, and as it turns, it catalyzes an addition of phosphate to ADP, as you can see here, and it captures that energy from the gradient as ATP. So here's the net overall of cellular respiration, ATP formation. So 1 FADH2 is equivalent to 1.5 ATP, and 1 NADH is equivalent to 2.5 ATP. Here are the steps of cellular respiration, again, in summary. So glycolysis forms 2 ATP and 2 NADH. 2 ATP goes to 2 ATP, 2 NADH, we multiply by 2.5, and we're left with 3 to 5 ATP. I'll explain what that is momentarily. Pyruvate oxidation, we're left with 2 NADH. Multiply that by 2.5, we have 5 ATP, so on and so forth. Krebs cycle, 2 ATP, 2 ATP. 6 NADH, 15 ATP. And 2 FADH2, 3 ATP. And if you add all these numbers together, you should get between 30 to 32 ATP in total. So the reason why I put an asterisk where the NADH forms in glycolysis is because NADH is made in the cytosol in glycolysis. And the NADH molecule cannot cross the inner mitochondrial membrane to deliver the electrons to complex 1. So the electrons have to be delivered via shuttle system, and this leads to reduced ATP production. So in summary, we started with 1 glucose molecule, and we underwent glycolysis, pyruvate oxidation, the Krebs cycle, and oxidative phosphorylation, and we're left with our net total of 30 ATP. Thank you very much.